Hi, everybody, and welcome to a summer school edition of Biology Bios. So this one is going to be a bit different. At the suggestion of a good friend, Dr. Jaime Sabel, I invited Dr. Judy Cole onto the podcast to give a perspective on her career, answering my standard questions, but at a different career stage than a lot of folks that we've had on. Unfortunately, Judy passed away a few weeks ago in June of 2025, and she was a dear friend, an amazing colleague, and a wonderful mentor. And I took a while editing this one. It seems I kept finding other things that needed doing before I did this, but it was a pleasure to come back to and to hear her voice. And she jokes a couple times that I'm going to have trouble editing this one down, but the joke's on you, Judy, because I did hardly any editing, and that means that y'all are in for a really long episode. It's about an hour and 15, hour and 20, but I hope you enjoy it. Yeah, here's Dr. Judy Cole. So, I'm going to welcome Dr. Judy Cole to Biology Bios. Thank you so much for being here.
Well, I think, Jim, you know perfectly well that nothing I like better than talking science. Not so much about myself, but I really loved what I do.
Well, I'm going to make you do both today.
Ah, okay, fine.
So yeah, generally I have people on, and I say, give me like the broad brushstrokes of your interests and what you do, where you're at currently in your career, and then we can talk about how you got there.
Okay, cool. Well, I am not in my career right now. I decided that for personal reasons that it was time for me to step away. My last graduate student finished during COVID, and I had no desire to write more grants. And I really hit the mother lode with my last graduate student, who was fabulous. And I thought, I think I'm just going to lean back, maybe write some papers I haven't finished, and just focus on teaching the last 4 or 5 years. So I retired in December, and I am reflecting on all of the things that have changed in science since I started out.
Yeah.
So I grew up in New York, and it's not a lie when people tell you that they're in a particular scientific discipline because maybe a family member has heart disease or diabetes, or they had a teacher or a multitude of teachers that influenced the direction that they took. And I can tell you right now that my high school biology teacher, Bill Fraunheiser, was the reason I went to graduate school in a biological discipline. Now, I thought, and this is true for a lot of people, I don't know why it's true for women so much, that we thought marine biology was what I should do. So I looked for schools where there was a big marine biology program. And the University of Rhode Island, first of all, it was far enough from home that I would have to stay on campus. Big yay. And the ocean was really close. And their biological station had a research vessel. Bob Ballard is now a professor in residence there, the guy who found the Titanic.
Oh, wow.
And I thought, God, this is just what I want to do. And then I took my introductory marine biology courses and said, I don't think so.
What was wrong with marine biology?
Nothing as a discipline, but the supporting courses just didn't interest me.
Huh.
So I decided to be, at biology at the University of Rhode Island is both a Bachelor of Arts degree at the time, and to get a BS, you needed to focus on a discipline.
Okay.
And so you could get a BA in biology or a BS in micro, zoology, botany, or ecology. But you still had to take classes in all of those disciplines. And no offense to my ecology-minded friends, it didn't blow my skirt up, as my grandma would say. But zoology, and particularly physiology, just was fascinating to me.
Cool.
And really made me become, well, let me rephrase, push me down the physiological side, was being a zoology major, and then a professor, Dr. Robert Hill, whose presence in the classroom and in the lab just was stunning to me. His interest was and love of the field was contagious to the point where I just wanted to be in the lab all the time. I can tell you now what he looked like. I can tell you right now what he wore to class every day. I don't know if he had multiple versions of this outfit or not. White shirt, black tie, gray pants, pleated with cuffs. And he would ride to the building on his women's bike with a basket in the front. And that's where his briefcase would be. But he made physiology so interesting. And the labs that he had developed were, well, today we're going to measure the intensity of skeletal muscle contraction. Here's how to do the dissection and go.
Ah.
So we made all of our own buffers. If we had a problem, we had to figure it out.
Yeah.
And for anyone who's a scientist, you know that your problem-solving skills are among the most important things that you learn. And that's where I learned, well, what have I done wrong? Oh, the recording bridge is on upside down. That's why it looks like that.
So many steps, though, that where you can get that wrong. So you got to get
that's
through
exactly
each
right.
of those.
And I learned the importance of a lab notebook in those days. So when I was thinking about what to do next, and I'll be honest with you, I had never thought about going to graduate school because, like pretty much anybody who's got a biology or a biochemistry major, let's go to medical school.
Yeah.
Here's my problem is that I really don't like sick people. And I knew that soon enough that, I mean, I did apply and I got as close as a waiting list at a couple of schools and then just said, this is really not for me because I wasn't disappointed that I didn't get in.
Right.
So I took a gap year, worked just a regular non-science job and said, I'm going to go get a PhD in physiology.
What was your non-science job?
I was working in the invoicing office in a Walmart-like shop in New York. I had math skills, but that was boring work. Drove me back to academia. But I also didn't have to worry about being a salesperson. So yeah. I was just there, receiving stuff, writing it came, or we're missing an item, and then sending out invoices. Uh-huh. It was a job. I made some money, paid back my student loans, which in those days were not nearly the burden that people carry today.
Like in a year, you made back your... Wow.
Yeah, I did. But again, college...
That's different.
Very different from where people are right now. I totally know how lucky I was with that. Because my parents were not wealthy people. I was one of those people who got one of the first Pell Grants as well. Nice. Because, believe it or not, Claiborne Pell was the senator from Rhode Island, and I felt pretty proud that I got one of the first ones. That was nice. Okay. So I didn't really want to move too far, well, first away, too far from the Northeast, and more importantly, not very far from the ocean. Yeah. So I looked for good physiology departments on the East Coast, and I ended up at the University of Connecticut. And I ended up in a comparative physiology laboratory with Nancy Clark, whose interest was in the regulation of calcium and phosphate metabolism in pretty much any non-mammalian vertebrate that walked in the door or slithered in the door, because we did snakes and birds and we had seagulls. But the long-standing model in her lab were domestic chickens and then starlings. Now, starlings are not beautiful birds.
Oh, I think they're great. They got that iridescence.
Well, but they are wonderful and they're incredibly smart and really good study subjects. I don't do animal work anymore for a variety of reasons. First, I had to catch my own starlings.
That's hard with a smart bird.
It is, and getting enough in the summer months when they don't flock is really hard. And so the study I was going to do was related to seasonal changes in parathyroid gland production of hormone and the structure of the gland. If you're not using it in the winter, does our structural anatomy change? Yeah. So we did a electron microscopic study, and we did physiological outputs for parathyroid hormone. Now the problem is we had no assay for circulating levels of parathyroid hormone.
What? So, all right. So, quick question on parathyroid hormone. Mm-hmm. Why parathyroid hormone?
Well, because
what's so great about parathyroid hormone?
It is the singularly most important hormone regulating calcium and phosphate homeostasis. Okay. And in terms of therapeutics, It is a singular compound that induces new bone formation. Now, there are a lot of drugs that prevent bone breakdown.
Right.
Is only one, well, maybe two, that promote bone growth.
Okay.
And so the drug companies were very interested in finding a way to use parathyroid hormone to promote bone growth. And I have to tell you and be upfront about this, I got my PhD in 1985. So not only were there not good models, there were no tools. And
especially not in starlings, I imagine.
Yeah, so there wasn't even an assay for parathyroid hormone in humans.
Oh, wow. Yeah.
So to try and figure out what parathyroid hormone levels were like in birds, to do those correlations, we had to find an assay. And that was one of the first things that I did, was develop an assay that measured cyclic AMP levels in bird urine. Now, I can see you all going, bird urine, that's not a thing. Well, it can be. If you treat the birds in a particular way, if you volume expand them a little bit, they'll make liquid urine. And then you can solubilize some of the particulate matter and get calcium out of that as well. But it turns out, and this is true in mammals, humans, birds, I don't know that I ever looked in reptiles because I wasn't working in reptiles, but parathyroid hormone, at least as far as I know now, I haven't read the PTH literature in a long time, it's the only hormone that stimulates the secretion of cyclic AMP into the developing urine. So if PTH levels are high in the blood, cyclic AMP in the urine would be high. And so what we were able to do, really instead of the seasonal study, was correlate changes in calcium and phosphate with changes in cyclic AMP excretion in the urine, and have a profile of how the levels of those compounds changed as parathyroid hormone levels changed.
Okay.
And it's not a direct assay, and as far as I know, there still is not a direct assay for non-mammalian forms of parathyroid hormone.
Interesting.
Mostly because it's not as important in chickens. We don't necessarily care.
Yeah.
We do care in those days, birds were excellent models because the calcium stresses required to lay a calcified eggshell are very high.
Right.
And so they turn over their bone very rapidly, and they make a calcified eggshell.
So if you're interested in how to grow new bone,
exactly,
look in a critter that has to do it all the time. Right.
And so there was actually two aspects, graduate students working on the excretion of calcium and phosphate, and then the impact on bone in birds. And I think you all know that bone in birds is really different than it is in mammals. It is very light.
Yeah.
And that's because they have medullary bone. So they don't fill up the marrow cavity with bone. They fill it up with spicules of bone that can turn over really quickly. So we were looking at those two really important targets for parathyroid hormone. And the other thing that's important about PTH is it makes sure that if you're retaining calcium, so it promotes the kidney's ability to retain calcium, but it causes birds to excrete phosphate.
Okay.
And even more importantly, in domestic fowl, it stimulates phosphate secretion. Well, isn't excretion and secretion the same thing? No, because birds can actually put more phosphate in their urine than the concentration of phosphate that's present in the blood. So they are actively moving phosphate from the blood into the urine. So sometimes twice the amount of phosphate is in the urine as can be explained by its concentration in the blood.
Wow.
Well, big fat hairy deal. Why do they have to do that? I'm sure there are people out there who've tried to add calcium to a phosphate buffered saline and failed miserably. It's because calcium and phosphate together will precipitate out a solution. And that would happen in your blood if you broke down bone, you release calcium and phosphate at the same time, and you have to get rid of the phosphate.
Okay. So, all right, you're mobilizing your calcium out of your bones. If you don't dump the phosphate, you're going to basically clog up your blood with these little white flakes.
Well, yeah, and they, lead to kidney stones and stuff like that.
Yeah.
So that's an important physiological role for parathyroid hormone, besides retaining calcium and promoting bone formation, is to get rid of that phosphate.
Yeah.
Mammals don't secrete phosphate, but they still enhance their excretion of it in response to the hormone.
Okay. Well, I guess that makes some sense that we wouldn't have as dramatic, rapid upregulation of a calcium system like that, right? We're not making a hard eggshell for our offspring, laying a new one every day if it's that season, right?
Right. Even chickens have to take a break. They don't. Yeah. They don't lay 7 days a week, and they don't do it for their whole lives.
On the seventh day.
On the seventh day, rested.
Okay, so that was your graduate work. You're going down this physiology trail, and then you took a little. You stayed in parathyroid hormone, yeah?
Yeah, because my postdoc, this is probably the most fortuitous postdoc acquisition ever. I was writing my dissertation. I hadn't started looking for postdocs. I probably should have started earlier, but remember, this was 40 years ago. And postdocs were easier. Funding was 1 grant out of 3 got funded.
Wait, I'm sorry. Go back, please.
One grant out of 3.
I don't understand what you're saying.
So money was not easy to come by, but substantially easier than it is today.
Sure.
And Nancy had a long-standing track record with NSF. So at the end of a granting year, we would often have, I don't know why she would save money back, but that's how we got our fluorescent microscope, because she had some money left from a granting year. So I had actually interviewed at the University of Connecticut Medical School, and it was a bone biology postdoc, and it was PTH, and it was bone, so I was still in the same hormone of interest. I wasn't excited about bone, but I was willing to do it and switch gears a little bit. And then Leonard Fort, who was at the University of Missouri School of Medicine in the Department of Pharmacology, called my advisor and said, "Got anybody finishing?" And he was the basis of a lot of the work that led to my assay development and my understanding of calcium and phosphate homeostasis in birds. And he wanted to know how to do some of the things that Nancy did routinely in the lab. And she said, "Well, this is a fortuitous call. Judy's going to be done in about 3 or 4 months." And that's how I got my postdoc. That's
pretty fantastic.
That's also a surprising thing to say in this day and age, that someone called and asked my mentor if she had anybody finishing.
Well, though, I just got an email from a colleague. I mean, he blasted it out to a bunch of people.
Right.
And said, "I have a postdoc who's leaving. This project still has funding. Do you know anybody?" So, I mean, people are still going out to their networks. I think it just looks a little different.
I think that's fair, because, I mean, I got what I thought was a substantial amount of money, considering my graduate stipend in the second most expensive state in the country was insubstantial.
And then you get to go move to Missouri
and make 3 times as much money, which was still, when I look back on it now, man, how did I live on that little money? But again, it was 30 years ago.
Yeah.
So anyway, and what I learned to do, I mean, I taught them how to do the things I knew how to do. But really what interested me was the pharmacological aspects of what Leonard's work was. He was looking at the upregulation of that's pharmacology speak.
I mean,
it's the increase in the number of parathyroid hormone receptors. We call that upregulation because we need a word for it. That was caused by estrogen levels changing during the course of eggshell formation.
Oh, yeah, okay.
So he wanted someone who knew the assays and had the skill set to do the renal function assays in chickens.
Yeah.
And I wanted to learn how he measured receptor number and affinity for ligands.
Yeah.
So it was really an incredibly good match. And what it did was turn me from a physiologist into a pharmacologist, looking at regulation of receptors and mechanism of, I don't want to say drug action, because that's what pharmacology makes people think about when they hear pharm, which means drug. But it's really mechanism of hormone or drug interaction with the target in the body. So that's what I learned from Leonard, and that's what made me a pharmacologist, really a receptor biologist.
Okay. Yeah.
And I think that's what got my job at the University of Memphis, was the importance of that, not just in understanding endocrinology or drug mechanisms, but for those things like we were talking earlier about therapeutics tamoxifen, which is used to treat breast cancer, is an estrogen-like molecule in some tissues and antagonizes or blocks estrogen receptors in other tissues. So it binds to the estrogen receptor, but depends on what receptor that's expressed, whether it turns it on or turns off as estrogen responses.
So this is an ER alpha versus ER beta
Absolutely,
thing, yeah?
Yep. And that's true for a lot of steroid hormones. Glucocorticoid has multiple receptor types.
Yeah.
Testosterone, but in the 80s we didn't know that.
I can say things like ER alpha and ER beta with some like authority because I worked in a steroid hormone lab for a while before grad school. And that was just at the time that, now we know that there are actually some membrane-bound steroid receptors.
Yes, and that's a relatively recent.
But we didn't know that.
No.
Then. That we had a postdoc in the lab that was like, I think that this is happening on the membrane, and everybody's like,
everybody laughed, didn't they?
Yeah, but then he did the work, and lo and behold, there's a progesterone receptor that seemed to be active on the membrane.
Well, I'll tell you, when you have some of those kinds of experiments that are arguing against dogma.
Yeah.
And I actually gave a talk, which was not really in the realm of possibility at the time. I went to a meeting to talk about protein kinase C being a potential second messenger system for parathyroid hormone.
Okay.
And the first question was, the preamble to the question was, as you well know, cyclic AMP is the only second messenger that's been defined for hormones and neurotransmitters. I said, well, clearly not, because it's clearly possible to elevate calcium and activate protein kinase C. Well, that was, anyway, it was an interesting talk at a national meeting, and nobody passed out and died, but nobody actually really believed us. But we had very good data.
Yeah.
And the next thing that was really different was the understanding that parathyroid hormone could activate the epidermal growth factor receptor. That was antithetical.
So it could actually bind to that receptor?
No. What it does, and here's really something very cool. One of the first things you do when you burn yourself or cut yourself, especially if you're a little kid, you stick that finger in your mouth.
Yeah.
There is epidermal growth factor in your spit, along with other things that are wound healers. And so what is interesting is that any cell that has epidermal growth factor receptors also makes epidermal growth factor or similar molecules.
Okay.
And so what parathyroid hormone does is stimulate the secretion of EGF receptor-like molecules.
Okay.
And that's how it activates the EGF receptor.
Cool.
Very cool. And it's probably the point in my postdoc where I decided I wasn't going to look at calcium and phosphate anymore. I was going to look at signaling and crosstalk between G protein coupled receptors and growth factor receptors. So a lot of times you reach a point in your career, particularly in academics, where it's easier to make these shifts in your scientific area. That's where it happened in my lab. Was this is too interesting not to follow up. So I started looking at signaling, particularly crosstalk between growth factor receptors and G protein coupled receptors. And it goes on a lot, and it's common for many G protein coupled receptors to do it.
And is it similarly a downstream effect of activating the one receptor produces something that then interacts with this other receptor?
So it's an autocrine loop, basically. PTH activates its receptor, which stimulates the production of EGF-like growth factors and then activate.
Yeah.
Because you can show easily that parathyroid hormone, under certain circumstances, particularly in epithelial tissues, promotes cell growth.
Yeah.
And no one thought that G protein coupled receptor hormones promoted growth. Well, I will tell you that the name of the things that promote the growth of gonads are called gonadotropins, and they're all targets for hormones that activate G protein coupled receptors. So yes, they promote growth. Just look at the name of the compounds.
Yeah.
So again, there we were knocking on the door with people who were saying, well, everybody knows that G protein coupled receptors do not promote growth. Well, clearly they do.
There was I'm going to get it wrong who said this, but it was in a crossword puzzle a few months ago. It might have been Carl Sagan. That was like, the most interesting thing in science that can be said is, oh, that's odd. Or something like that. But like yours is kind of the flip side of that coin is like, if somebody's telling you, well, that can't be, that's just another way of saying, well, that's curious.
That's right. I remember, I don't even know how long ago it was. It was a commercial from a scientist who worked at maybe Lilly or Park Davis. And she said, the best days in science is when you get data that are just the opposite of what you expected.
Yeah.
And you're the first person who sees it. And really, that was the most exciting day. It was when I was a postdoc, and it changed the direction of where my science was going to go.
That is super cool.
Very cool. So I went from being a comparative physiologist to a cell biologist to specifically a receptor pharmacologist and signal transducer.
So, okay, so we've dove into some serious. Well, for me, so as a bird chaser ecologist, that felt pretty like hardcore molecular to me. But for hardcore molecular people, it probably wasn't. But I was going to ask you. So that's clearly like a pivotal moment. Looking back at maybe projects since there. Is there a project or maybe a couple projects that you're particularly proud of that like, man, that was the one, that's my highlight?
Well, I will say in my postdoc that, let me explain to you how we shifted gears. There was a graduate student down the hall looking at the platelet activating factor receptor, which is a G protein coupled receptor. Mm-hmm. And what he was interested in was getting a negative control because he had some evidence that platelet activating PAF was activating a tyrosine kinase. And I said, well, I can give you some parathyroid hormone because it doesn't do that. And he came back 2 days later because the way that you looked for tyrosine phosphorylation was using gobs of P32 and looking for changes in the amount of phosphate incorporated into a protein.
Okay.
So he came and showed me the autorad, and I said, well, clearly you made a mistake.
So, all right. So for the
For the uninitiated?
For the uninitiated, P32, radioactive.
Right.
In the autorad, you're looking at how much radioactivity actually got incorporated, right?
That's exactly right. And you do it on X-ray film so that the denser the granules are in that X-ray film, the more P30 phosphorus.
Yeah.
And we already know that bone is a really important storage site for phosphorus. And he came down and he showed me in the PTH-treated cells, there was a marked increase in tyrosine phosphorylation of proteins. I said, well, clearly, Archie, you didn't do the experiment correctly. He said, well, clearly, Judy, you didn't give me parathyroid hormone. Well, I took umbrage at that because he did not have a PhD and I did. So I gave him some more and he did the experiment again. And guess what happened? The same damn thing.
Yeah.
We did it a third time, same damn thing. I said, I believe I need to follow this up.
Yeah.
And that changed the direction of what I wanted to do in the future. A single experiment when I was a postdoc changed the things that I was thinking about.
Sure.
And when I first got here at Memphis, there were maybe 2 people who were interested in signaling. I liked them both, but they were not signalers, in spite of what they would say. I will not name names. But I actually started working on a project with Tom Sutter, who is a toxicologist. And one of the things that changes in response to exposure to dioxin is that it causes the production of TGF alpha, which is tumor growth factor. And that's an EGF receptor-like ligand.
Okay.
And I was an EGF receptor ligand kind of person. And it turns out, so this is a very nice series of studies done by a graduate student who looked at the ability of, first of all, was dioxin directly activating the EGF receptor? And the answer was no.
Okay.
Was it working through TGF alpha? And the answer is yes. But in skin makes at least 3 types of EGF receptor ligands. And each one of them subserved or promoted a different function or produced a different function that dioxin caused. So TGF alpha regulated the receptors differently than a compound called heparin-binding EGF-like growth factor, and also amphoregulin. And each one of them modified the responses to dioxin distinctly different because it modified the EGF receptor activity differently.
Okay.
So that was among a series of really cool receptors.
Are those like cofactors for that receptor, or is that
there are
something else?
ligands produced by the keratinocyte itself. And really, cells don't do things just 'cause.
Yeah.
It's easier to use the same receptor and make multiple target molecules than it is to make multiple receptors.
Yeah.
Because, the EGF receptor is a big protein, and it takes a lot of energy to make lots of protein. But if you make a ligand, a hormone, a growth factor, that's subtly different in the structure of the receptor when it binds, you can get a different subset of responses. So it's a really energy-efficient way of producing different responses through a single protein.
Right.
And it's not uncommon.
Yeah.
So that was a really important series of experiments that I'm really very proud of. And the graduate student who did the work really did a nice job. She was very smart. We didn't use radioligand binding anymore because I would prefer not to use radioactive iodine in the lab.
Oh, you were using iodine?
No. Because of my assay background, we decided to use a modified EGF molecule.
Okay.
So we bought a biotinylated epidermal growth factor and then detected its binding with an antibody to biotin. So it was a fabulous indirect way of measuring changes in the receptor.
A heck of a lot better than messing with radioiodides.
Absolutely. And what we discovered was, depending on the ligand, the amount of receptor on the cell surface changed.
Uh-huh.
And if you don't have receptors on the cell surface, you can't produce a response.
Right.
So having the multiple ligands could preserve the receptor through TGF alpha, but downregulate or reduce the number of receptors in response to amphoregulin.
Cool.
So you could keep things going in this response element and prevent responses on the other.
Yeah.
Very, very cool. Very cool work.
Man. Okay, so I'm gonna go through. I've got my list of general questions here, and I think you already hit on this. But maybe I should ask it explicitly. Was there a time that you knew that you wanted to be a scientist? You talked about these people that were really important along your scientific journey, but was there, similar to that moment in your postdoc where you're like, I'm gonna chase that question? Was there a moment where you're like, I'm gonna be a scientist?
Well, I'll tell you. What it is for me is when things happen, I wanted to know why.
Yeah.
I will tell a brief story about, what the problem is with scientists? It's that when you do something, you go, I wonder if that would happen again.
Yeah.
Now, that's really okay in the experimental lab, but it's not when you stick a fork in an electrical outlet. So I was curious, and so I went, I wonder what would happen if I did. Well, I found out what would happen if you stuck something in an electrical outlet. I guess I'm lucky I'm still here.
How many fuses did you blow?
Well, that's exactly right. I think it was only one. But I think that's what it is. It's, I wonder what would happen if I did this.
Yeah.
And most of those, I wonder what would happen, were all science related. Well, how come this happens when I do that?
Uh-huh.
And then I picked biology because I liked trying to understand why things happened. And what I mean by that is, well, why if you drop a rock on your foot, does it hurt?
Yeah.
Or how come you don't have to think about pulling your hand away from a hot fire? Mm-hmm. It's because you don't have to think about it. And I knew when I went to college why that is. You have reflex arcs. You don't have to think about pulling your hand away because your brain doesn't get involved.
Yeah.
But I didn't know that. My mother just said, don't touch a hot stove, and then I wanted to know why. Mothers are responsible for most childhood injuries, I'm telling you right now. And that was the case in my house. It was, I don't want you doing that. Why? And so then I would go do it. I guess I'm lucky to be here.
Did you ever see, this is totally random, did you ever see the Beatles Yellow Submarine cartoon? There's a thing where there's a lever, And I think it's Ringo. And they're like, don't pull the lever. And he's like, I can't help it. I'm a born lever puller.
There you
I'm
go.
a born lever puller.
It's always I wonder why.
Yeah.
And I think that's really what it is. And because biology interested me.
Yeah. Yeah. I think it might have been Steve Nowicki when he was on a few weeks ago said something similar, that like there's also an immediacy and a relatability to biology that, like, I mean, I think about the stuff that like astrophysicists do, and it always makes me feel a little bit small, because I'm like, well, that's a big question. You're talking about, millions of light years away. Like, that's huge questions. But like for my brain, it's maybe a little too huge.
Yeah, I agree.
It's like my brain likes the things that are more like, oh, okay, so that's why that thing happens in my body, or that's why that critter appears in that area.
One of the things I like about, regardless of whether it's wet lab or field biology, is the feeling you get when you go, I'm the first person who's ever seen this. And that does happen still in this day and age.
Yeah.
I felt that way when the EGF receptor was phosphorylated. Oh my God, there's nothing about this in the literature anywhere.
Yeah.
Which gives one pause, first of all. Well, maybe it's an artifact. Archie, are you sure you did this experiment right? But, being the first person to look at your data and go, no one's ever seen this before. Yeah. How exciting is that?
Yeah.
I guess that's what the old explorers would sail their ships to do, is to find the new route to China or India for tea. You're the first person to do it.
It's a pretty unique, like, dopamine hit.
Pretty unique. You know how I feel about modifying unique.
It is a unique dopamine hit.
Oh, I think that's absolutely true. It's biological and it's chemical, and it's feeling of dopamine is really a fabulous molecule. It makes you feel good when something really positive happens.
Yeah.
And I think biology, when you do your experiments, that's the feedback you get.
Yeah. Yeah. That's pretty meta.
I know, right?
Okay. So really big right turn from like hardcore science to the sort of more human side of science. And I preface this with people that, like, we get a lot of negative feedback, and not in the sort of receptor antagonist way, but like from the outside, people telling us, well, you should have done that experiment, or this grant is not up to par. I also certainly generate negative feedback and second guessing myself and thinking, well, that experiment that you planned out, that's maybe not the best, or I'm meaner to myself than that. But like, how do you, and how have you over the course of your career confronted that sort of internal negative feedback?
Well, I totally understand that, because one of the first problems I have, everybody says this, it's less true than it was, is two things. You always hear the thing about imposter syndrome.
Yeah.
That you're not good enough or smart enough, and I'm better about that. My second problem is I want everything I've done to be perfect. And what I mean by that is I want to have thought of all of the questions that reviewers will ask and try and preempt their comments. And so I have ended up keeping papers too long.
Yeah.
Before I send them out. Yeah. So the answer is I in the past probably 10 years, I've gotten better about, I actually am pretty good at what I do. Because at least in my department, when there is a question about receptors and signaling, I have people come and talk to me all the time. Bioengineers have come and talked to me about the best way to grow their cells. Well, it's not on titanium, unmodified. You really need to give them... So basically ideas like that, where other people appreciate what I do more than I do. And the other thing that I do is I am better about letting go.
You mean letting go like those papers?
Letting a paper go out.
Yeah.
Because sometimes, not always, it's always the third reviewer who is problematic, but the other reviewers give you some insights and things you didn't think about.
Yeah.
And I, in most circumstances, will tell you that the reviewers, if they're like me, which most are, they give you feedback that will make the paper better.
Yeah.
And that's been, for the most part, my experience. Have I been mad? Of course. I'm particularly disturbed when it's clear to me people haven't read the paper. And it's really disappointing when it's a pharmacological paper and they don't have a pharmacologist read it, because I've had people say, well, you shouldn't have used this drug because it's not as potent. Well, that doesn't matter. It's not how much or how little of a drug you use, it's how well it works.
Yeah.
So if a drug works better than another, it shouldn't matter how well it binds to the receptor. As long as it's not toxic at high doses, you pick a drug based on how well it works.
Yeah.
So that was a hard review to answer without getting all pissy about it.
Yeah.
So
this was something that you're looking at, like more of a whole organism response or a system response.
Well, at least
to
in a
something?
cellular response.
Okay.
You should have used this drug because it takes, the potency is twice what the inhibitor was that you used. I said, yes, but it has off-target effects.
Yeah.
And it's not as effective at producing that response as the one I chose.
Yeah.
And that's really important. And if a pharmacologist had read the paper, they wouldn't have had a problem with my choice.
Yeah.
And so when I review papers, that's what I try and do is, well, what you really should do is think about it this way. That and people who don't use the proper controls are my biggest complaints.
Wow. This is so, all right. So coming from the ecology side, like we often can't do all the proper controls, right?
No, I understand all that, but to use an inhibitor that's supposed to be selective and then not asking, well, does it affect something that's similar but not identical to it?
I'm always really in awe of the way that people that do molecular biology like you do design their controls and think so hard about the controls. Because really, at the end of the day, I think that's what makes the experiment super good. Because like you have, all right, I'm interested in this thing. Obviously, you're going to give that thing as a treatment. That part's easy.
Right.
But all the other controls that go, like, people pride themselves on that rightly, I think. It's a very different thing that I've been trying to get better with my sort of like blend of field and lab stuff, but...
Well, I will tell you that any graduate student upon whose committee I've sat will understand that. Because really, if you are making an argument for a particular compound to work in a certain way, you really need to show that it has no or limited off-target effects. And I know that I've frustrated graduate students, but really, you have to publish it.
Well, yeah. I was just going to say that's exactly what you said about the reviewers. That you can't think of everything.
No, you
as
can't.
Good as you are at controls, there's some damn reviewer that's going to think of a control that you're like, oh crap, that one's really good.
And I appreciate that kind of feedback because all the experiments may not be simple, they clearly should be done. Yeah. And I almost inevitably thank the reviewers for that insightful comment. Yeah. So when I do a review, I'm very careful about the language I use because my goal is to support the authors getting that paper out. Yeah. Most of the papers I review, have I outright said no to papers? Absolutely. Yeah. Or needs lots of revisions. But I just finished a paper that I thought was really, really good. But it was a pharmacological concept that lots of people misunderstand or don't describe accurately. Their data clearly showed what they meant, but they didn't explain how they got there and why their experimental design proved that. And I really wanted them to do that because it was the best example I've ever seen, I'm going to tell you, of synergistic responses. Ooh. And synergism is not a simple concept.
No. I need to talk to you more about that. I do, because we've been thinking about synergisms in a statistical sense.
And you really do need to think there are actual formulas that help you make that decision. Yeah. Whether it's additive or competition or synergistic.
Yeah. Yeah. Okay, so let me pivot to advice here and think if you and you've advised so many graduate students and undergraduates, but thinking kind of your own path in biology, if you met that undergrad version of Judy Cole working in today's world of biology, what advice would you have for that person?
I wish I'd talked with my professors more.
Okay.
And I'll tell you, I like it when students would drop by and chat, mostly because I was doing stuff in my office that didn't interest me. But I love talking with students.
Yeah.
And I think students, particularly in really big institutions, now URI was a state school, but there are like 12 people who live in the state of Rhode Island. So that school, we had 7,000 undergraduates at that time. It's not like the University of Missouri with 50,000 or Michigan, I don't know, what do they got, 80,000 or something?
I don't even know.
So, and Memphis is fairly large. And I try and tell students to come and talk to me because I love to talk to students. Faculty, for the most part, my colleagues everywhere like talking with students, but we don't go out of our way to get them to come.
Yeah.
Or encourage them every day to come and talk. Talk to your professors because we're lonely. And we really don't want to be reviewing that paper. And we don't want to go to that committee meeting. And we don't want to whatever. But we love to talk about what we do and encourage. At least I do. And those are the best times. So talk to your professors.
Yeah. And I've found over the years that like just a little, it doesn't have to be like over the top flattery, but you just come to somebody and you say, what you do sounds really cool. Mm-hmm. That's enough flattery that the person's going to be like, well, let me tell you more.
Well, I talked with Al Gilman, Nobel laureate Al Gilman, at a meeting. He had given a talk on X-ray crystallography. Uh-huh. He was out having a cigarette and gathered up all my courage and went and said, I just want to tell you that may be the best X-ray crystallography talk I've ever been to because I don't understand it, but you help me understand it. He said, well, then my work here is done. But, he's a Nobel laureate. And he took a few minutes just to chat with me. And it took me 15 minutes or so to just sashay over and suck it up and go talk to him. Yeah. I think talking with your colleagues, I think talking with graduate students, just talking back and forth. Some of the best advice I've gotten for experiments come from people who don't work in my field. Yeah. Because they said, well, have you ever tried this? No, but give me a pen. Yeah. That's an approach I'd never thought of. Yeah. So I think we don't talk enough amongst ourselves. We sure don't talk to the public enough. For the public to understand what we do. Now, I talk with my family a lot. Yeah. And I think you can see, and I don't want to go down a road too far, if we as scientists had been better about talking what we were interested in and why science was fabulous, COVID might have been better. Because the science during COVID was amazing.
Oh, sure.
And I talked to my family about how amazing the breakthroughs they made were happened to be. And we missed an opportunity to really, really educate the public. And we've been not educating the public for a long time now.
Well, yeah, I feel like that is a good example. And it is something that happened after many years of misconception of what science is.
Yeah. 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 We need to be better communicators to the non-scientific community because people are interested in stuff.
Yeah.
Yeah. I wish we had done better all along talking about how fabulous this is. Yeah. But you and I both know that making what we do approachable to people, first of all, who don't work in our field. So we just had that experience here. When I start talking signaling, I have to remember not everybody knows all the acronyms. Yeah. Yeah. Or is as thrilled with my data as I am. So even among scientists of different ilk, it's really hard. But to try and talk about what I do to people who don't really work in science, trying to find a way to explain it in approachable terms is really hard, but we should do a better job.
Well, I think, again, maybe this comes back to the distinction, and I'm not hating on astrophysicists. I really respect what you guys do, but holy cow, it is so foreign. Yeah. Like very literally very far away foreign.
Yeah.
But we have this advantage of like, all right, you know what happens when you touch a hot stove? Well, let me tell you more, right? Like
Exactly right.
This immediate in that, yeah, but there is a lot to navigate there.
Yeah.
And I will say that people do the physics thing well. Like you can do all of these things well. You have to be very conscious of it.
Yeah.
And I mean, we were geeking out and like we didn't define every term. We talked about G-coupled proteins or G-protein coupled receptors, but we didn't define those. And,
Right.
It's hard when you get on a roll.
Right.
I mean, I guess we could.
Well, I know my family want to hear it.
Yeah. Well, so Judy, why don't you define a G-protein coupled receptor for me?
I could do that.
All right.
And if I had stopped, would you like me to?
Yeah. I mean, I would do it, but I want to give you the opportunity. I would do it badly is what I'm saying.
You probably wouldn't. Here's the thing. I actually taught virology, 8 lectures of virology for Carlos when he hurt his back.
Uh-huh.
I'm not a virologist. I have a PhD in science. I should be able to at least convey important information to students about viruses and why they're cool.
Yeah.
Which, in fact, they are.
They are.
Trey cool. So I forgot where we were going. What was the question?
G-protein.
Okay. One of the things that's important about hormones and neurotransmitters and drugs is most of them can't cross a cell membrane. So if they have information they have to transmit to the cell, they can't get inside the cell to do that. Steroid hormones can.
Yeah.
But adrenaline, epinephrine, doesn't cross the plasma membrane. Dopamine doesn't cross the plasma membrane. They bind to surface receptors in the membrane, and then they have to, when they bind, change the structure of the receptor so it generates information. So the first signal is the binding of that neurotransmitter to the receptor, and the transduction event is the receptor conformation change.
So the receptor changes shape, and then something inside.
Right. So inside now, the signaling pathways recognize that, oh, that's the adrenaline conformation. I need to do this.
Yeah.
That's the dopamine conformation. I need to do that thing over there.
Yeah.
It's because dopamine and epinephrine or adrenaline are highly charged molecules, so they can't get across the lipid bilayer of the membrane. So if they have to change what a cell does, they have to do it by binding to a receptor. And the receptor signals to the inside of the cell through a G protein, which is either on or off, depending on the form of a G nucleotide binding site, whether it has GTP bound or GDP bound. And so the receptor conformation changes makes the GDP bound come off, allows GTP to bind, and the receptor becomes active.
So you basically got a switch.
You have a switch.
And very similarly, it's a change in, like, you didn't change what the light switch is if you flick a light switch. You changed what it looks like.
That's exactly right.
Yeah.
And that's the job of a G protein coupled receptor.
Sweet.
It is. Receptor tyrosine kinases also bind things that don't cross the membrane. They change conformation. They don't use a G protein. What they do is the conformation change activates an enzyme that generates a signal.
Yeah.
So that's how we get specificity in the response. So it's very cool. You can tell that I think it's very cool.
It is very cool.
And so I teach a class in receptor biology, and I tell the students on the first day, I said, you're already at a disadvantage. And then they all get all spooky and their eyes get big. I said, I started doing this stuff when it was easy, and there was 1 second messenger. Now I'm going to tell you about all the things that have changed since then and expect you to know it when I've accumulated the information over 25 or 30 years.
Yeah.
So that if you are interested in signaling, you're going to have a job forever, because every time you define a step, you discover there's another step down the lane.
Yeah. And this is like very much what you were talking about of those events where, well, we all know that this is, and by second messenger, like that's the receptor does its conformation change, and then you have these second messengers that actually
Tell
translate that into the cell.
Exactly right.
And we all thought it was all cyclic AMP at all the time.
Well, people didn't believe in receptors in the 60s, the 1960s.
Wow.
Well, what is it, magic? But getting people to believe in receptors was a huge hurdle.
Yeah.
Now, I was not doing science in the 60s. I just want you to be aware of that. But I read the history.
Still sticking forks in.
Well, yes, that's probably true.
Okay, so a couple questions left here. What do you do when you're not sciencing?
Ooh, God, I love listening to music. I don't play guitar as much as I used to. I like to read. I love sports. I don't participate much anymore, but during college football season or basketball during March Madness, I just like talking to people. So regardless of this podcast, I enjoy talking with all of my buds and colleagues in the department, and this has been kind of fun for me today because I don't talk science a lot to people who aren't science people.
Well, and you and I have talked about, it feels like everything under the sun at other times, and we have not gone a deep dive down receptors, which I think is kind of funny that like this has been like this huge part of your life, and like we are more likely to talk about who we picked in our brackets.
Well, that's exactly right, and I know you play guitar, and I do. Not as much anymore because I have carpal tunnel syndrome, and it makes it really hard. But there were some nights I would just come home and go up and get my guitar and just play for an hour to get the stress out. So, so music plays a big part of my life, and reading. I got 3 books on my iPad right now. I'm not reading them all at the same time, but I don't like to have to go and find the next book when I finish one.
Yeah, I don't like that either.
So I always have one in line, and then when I start that one, I go find the one that's going to be next.
Nice.
And then I listen to a lot of audiobooks too.
Yeah. Well, I often say, well, I've been rereading this thing, and what I mean is like I've been listening to it.
Yeah. Well, I have a friend who said, "You read books more than once?" I said, "There's some books I read every year. I used to read The Stand every year, Stephen King's Plague Book.
Yeah,
I got rid of my copies of the Stand after COVID.
That didn't last through COVID, no.
No, because it was prescient.
Yeah,
not quite as bad as what happened in that book, but I just can't have it around anymore.
Yeah. The one that I've been rereading now is re-listening to is David Quammen's Spillover. Which he wrote before COVID.
Yeah.
And he talks about SARS and like the potential of coronaviruses and bats and you're just like, oh my gosh.
Well, did you ever read The Hot Zone?
I never did actually. I mean, I know what it's about, but like
it was frightening.
Yeah.
How lucky the people were who had those samples of Ebola in the lab that it got out. Yeah. And if you all didn't know that, it did. But it was not the Ebola.
Yeah.
But it was, well, if you read it or listen to it on an audiobook, you'll hear scientists doing things that they should never have done.
Yeah.
Smelling the culture medium to see if it was contaminated. Never do that. It's like mouth pipetting iodine.
Good luck. Yeah.
But I read that book and just went, oh my God. That's a frightening book too. Okay.
So now I'm reading the Spillover thing. We could go down a whole frightening rabbit trail, but let's not. What else do you wish I asked you about that I haven't asked you about?
Well, not too much. We covered a lot of ground. One of the things that let me tell you what I'm most proud of. And while, I think I published or been an author on about 50 papers. And that's great. But in 5 years, most of those papers will not have any real important impact because the science changes. The things that I've seen since I started in science, you could get a PhD for cloning a receptor when I was a postdoc.
That was bloody hard to do.
Yeah, but nobody can do that anymore.
Yeah.
That's how fast things progressed. So the papers, while important, are not my favorite thing about my career. It's the students that I've turned out.
Yeah.
Because that's your legacy, really. And I will tell you that of the PhD students, I don't think there's any one of them who didn't go to a place with bigger and better toys. And that's really, I'm proudest of my students, because that's my legacy, where they are, what they've done, who they are, what kind of jobs they're doing. And I don't mean just my graduate students.
Uh-huh.
I got an email a couple of weeks ago from a student from 10 years ago telling me where he was doing his residency. 10 years ago.
Yeah.
And while, don't get me wrong, the papers are important.
Yeah.
What pleases me most is hearing from students, and it always surprises me, the things that they find important about interactions with me as a person. That's what I'm proudest of.
That makes good sense to me. And it comes back to, I wanted to say this when you were talking about talking to one another. And this is maybe a public misconception too, is that science is, or at least can be, maybe should be social.
Well, I agree.
And it's just having two pairs of eyes on a problem is better than one pair of eyes. Two brains is better than one brain.
Yeah.
I mean, okay, you can have too many cooks in a kitchen, sure, but like, we strive to understand truth. We strive for objectivity, and we do that through our experimental design.
Right.
But we're human.
Mm-hmm.
And I don't know. Sometimes I think that gets lost, and the vision that people have of scientists are these cold, lonely people in labs.
Well, that's true to some degree. I think people would say, when I tell people I'm really a shy person, they laugh. But it was hard for me to talk a lot early in my career. I was a listener and a watcher.
Yeah.
Now you can't shut me up because I love what I do, and I really think that people need to hear about that.
Yeah.
And I think most of the people I hang with feel that way as well.
Yeah.
And I really want students and postdocs and whatever to go away and be better than I was when I started out.
Yeah. Because
I could have done some things better that I didn't do. Speak up for myself better.
Okay.
Argue points better. As a good listener, though.
Yeah.
And I always went to stuff and listened, and had people who would come to my posters because I had written and done the posters. I like that interaction a lot.
Yeah.
So talking, really, I wish we had bigger group lab meetings. When I was a postdoc, all the receptor biologists in the department would meet once a week.
Oh, wow.
We didn't work on the same ligands,
yeah,
but we talked about receptors and how to answer questions and critiqued each other's work. But we are in biology, a very diverse department. Ooh, there's that word. In terms of the things we study,
yeah,
that it's hard for us to have a departmental kind of, I don't know, lab meeting or journal club.
Yeah.
But I don't think we ought to really think about that.
Yeah.
And I don't think we train broadly enough anymore. So that someone who worked in my lab can go and work at your lab with just a little bit of training on the things that are specific to immunology.
I mean, I think maybe not as a broad brushstroke, but I don't know. I feel like somebody from your lab could absolutely do the work in my lab.
And vice versa, because all you really, you know in immunology that cytokines have to tell a cell what to do.
Yeah.
I know how to ask what is the information that's being passed along.
Yeah.
People who are looking at reproductive biology have estrogen and testosterone to think about. How does the steroid hormones do that?
Yeah.
Well, we could talk about that.
Yeah.
And I think we should. Because I think a portion of talking about changes in behavior without asking how on the organ or cellular level, how that's working, and then couple it back to a change in behavior would be a great, complete story.
Well, yeah, that's the Tinbergen's 4 questions in animal behavior, right?
Right.
To really understand it, you can't just look at one.
Yeah, I think that's fair. That's fair. Not so that everybody knows how to do PCR, but understands what PCR is.
Yeah.
And what the output means in terms of information.
Yeah. Yeah. I mean, you're preaching to the choir here with me.
Oh, I know that.
I think even more broadly, you know, I like the liberal arts education model. Like I think it's really important that I feel like I play guitar and I write better songs when I'm doing more science, and I think that my science and my science writing is better if I'm playing more music. You know, I think this breadth is really important.
Well, I'll tell you that as an undergraduate, everybody has general academic requirements. I took a lot of literature and philosophy courses to fill out my Gen Ed.
Yeah.
Before I knew I was going to live in the South, I took a class called Introduction to the Southern Novel.
Oh.
We read Faulkner.
Yeah,
we read Flannery O'Connor. I'm not saying it was the best class, but it was.
Oh, no, that's good stuff, though.
Reading things outside
Yeah.
my area of major. Took a couple of Greek myth classes.
Yeah.
My father was particularly disappointed when I brought my philosophy books home. It was the philosophy of communism. And he was a World War II vet, and he was not happy.
Yeah.
But I think it gives you an understanding of what communism is, and I didn't understand what was so important about it.
Right.
And now living in the South, I understand what Flannery O'Connor was talking about, and Faulkner. God, Faulkner lived down the street.
Really? I didn't even know that.
Well, sure you did.
No. He lived in Memphis?
No. Oh, Miss.
Oh, okay.
You knew that.
Okay. I was like, wait a second. What? You pointed towards Midtown.
Well,
I was like, wait.
That,
He was
fair enough. And just along those lines. And so I think when people read To Kill a Mockingbird, if you've lived in the South now, you have a better understanding of what the problems were. Not 30, 60 years ago,
Yeah.
but understand why that was problematic.
Yeah.
I've learned a lot since I moved to the South about this country and our history. So,
Yeah.
and it was through my liberal arts aspect of my undergraduate degree.
Yeah.
So, did I take a lot of science? Hell yeah.
Well, yeah.
But I took a lot of things that many of my biology major friends would never have taken. Because I liked reading, and Introduction to the Southern Novel was reading novels. So I got to write a paper, big fat hairy deal.
I had similar interests, but I'll tell you, I could not buy an A in British literature. I thought it was a great class. It was super cool. We read Joyce and
Yeah.
a whole
You
bunch
read Joyce?
Well, we didn't read Ulysses and stuff.
Oh, I thought you were going to tell me you read Ulysses.
Oh, good Lord, no. But I thought it was super great. But yeah, I could not impress that professor. I was just like, well, there we go.
Well, I'm so sorry. I stayed away from classes where I had to read Moby Dick, though.
Yeah.
So I had to do that in high school. I didn't want to ever do it again.
That's fair.
Yeah,
That's
I think
fair.
so.
It's a lot of knots. Or a lot of wailing, sorry.
Yeah.
All right. Well, I think speaking of liberal arts education, we've done a pretty good job of ranging widely today.
Well, I think so. And over what's probably a long period of time, but doesn't feel like it.
No. I think this is going to be the longest episode of the podcast.
Well, you're going to have to trim some of it, aren't you?
I might. I don't know what I'm going to trim, though.
Well, we'll figure it out.
I liked it all. But yeah, Judy, thank you so much for doing this. This has been an absolute pleasure, and I think a really cool and different take on these questions.
Well, cool. I'm glad, because you already discovered this. I love talking about science.
Yeah, man.
And my niece, who is going to college pretty soon, I gave her the same advice. Talk to your professors, because they love to talk to students, most of them.
Yeah.
So I've thought about this a lot, about where I've been moving along in my professional life, and I have some answers already about things I might have done differently.
Yeah.
Starting along. So I've actually thought about this, because new faculty ask questions like that.
Yeah.
Well, what would you do under these circumstances? Well, not what I did. Let me tell you what I did that was just stupid as crap.
Well, shit. Now I want to ask you about this. One of those.
Well, we could talk some other time about things that I should have done better. But
are you sure? Because one nugget of that, is there one boy,
No.
I really wish I had not XYZ'd?
Well, it's more I wish I had done.
Okay.
wish I had been a little more forceful in my objection to some of the things that happened early in my career.
Okay.
The reason I left Missouri is related to the value that people placed on me as a faculty member.
Yeah.
Because I was non-regular faculty.
Okay.
I'm doing a damn good job. I had grant money.
So that means like a
non-tenure track position I was teaching. And the end result is that I left because there was no support for non-regular faculty. And a lot of women end up in that particular aspect. And I've had people take my ideas and put them in their own grants after telling me it was a stupid idea.
That's shitty.
It is shitty. But while some people think women in academics and in the scientific disciplines is much better than it used to be, that's not true.
I was gonna say, like, what you're describing is not gone by any stretch.
It's more covert. And what I mean by that is people are more aware that that's not acceptable behavior and modify their tone. But it's pretty clear it's still a problem.
And there are some places that do it better, like anything, right? There's some places that do this better than others. And one thing I'll say about Iowa State, where we left, is that they were very good in a lot of ways about non-tenure track faculty.
Well, that's good to hear because it's not been my experience.
Well,
and postdocs, regardless, are faculty when it's good for the university, and not faculty when it's going to cost them money.
Yeah.
And that makes postdocs sort of out there in limbo.
Totally.
Whether you're a guy or a gal, it's, well, you're a postdoc, you're not eligible for this. Well, but I'm an employee. Well, not really.
Yeah.
Unless it's valuable to us. So it's, having been both a graduate student, a faculty member, and a postdoc, things have changed a little bit.
Yeah.
And things are better. There's still a lot of room for improvement.
Yeah. I think what you hit on is this categorization of like, your position is this, therefore not worth, like, okay, if you have been doing a bad job at your position, if you have not been getting the money that you said you would get, if you have not been writing the papers you said you would write, if you've not been living up, okay, right? Like, right. There should be consequences, but that those are applied differentially. That's not great.
So I think, we're better than we used to be, but really we could be better than we are.
So, I mean, I think what you said you wish you'd objected more strongly or stood up for that more strongly.
I think...
But you're saying it now, which I think is really important.
Well, so that's okay, but it would have been better at the time had I gathered up all my inner resolve to say, this is not right.
Well...
there are now places where you can go and, at most universities, and really lodge complaints.
Yeah.
Would I have done that if I had had more resolve? Maybe. People do it more frequently now than they used to. But still, it's problematic.
Yeah.
So I wish I had been more vocal, but I don't know that I'd be here if I had been. And that's the rock and the hard place. So I can say what I want now because I'm an emeritus professor.
Yeah.
Or I can say what I want now. I have tenure.
Yeah.
And that really shouldn't muffle your voice if you don't have tenure.
Yeah.
And it does.
It does. I have no reference point to know what that's like in the private sector.
No, I don't know either.
I imagine this is something that is,
I think it's still problematic.
Widespread.
You reach a certain level and you don't go any further.
Yeah. Or you get past a certain level and then all of a sudden you're one of the people in the back room making the decisions.
Yep.
And that's...
Yep. So that's a real turn here in this otherwise uplifting conversation.
I mean, we could talk about what we want to include, but like, I think it's important for people to hear that when they feel that way, like other people have felt that way.
Oh yeah. It's not uncommon when women get together and talk. I think we have a lot more women faculty now than when I started here, and that's been such such a supportive system now for the women who were in the department. It was, when I got here, it was Dr. Barbara Toller and myself, and now we have 7 women faculty, and we're very supportive of one another in ways that I don't think that my male colleagues actually think about. But it's been really important to increase the number of women, not just for our own sake, but because 60% of our majors are women.
Yeah.
And so our classroom should reflect that.
Yeah.
Now, I think that we have a lot to say.
Yeah.
And our approaches to doing business are different than guys.
Mm-hmm.
Not better, not worse, different.
Yeah.
And I think one of the things that's nice in many science departments is rotation in labs.
Yeah.
Because people are maybe asking the same questions but are approaching it differently. And different approaches to same questions really gives graduate students and postdocs insights into, first of all, the variety of ways that you can ask questions, and then the way people think about questions. So if we do rotations, that makes that easier to understand.
Yeah. Yeah. So for any undergrad that's made it 2.5 hours into this, or however far this is, as a grad student, in a lot of programs, you'll do, say, 6 weeks or a couple months in one professor's research group, and you'll have some small project that, you think you can make some headway on in that time. And then you'll go to another one, mm-hmm. and then you'll go to another one. And oftentimes, like in the programs I've seen, then you end up saying, well, I really liked Lab B, and I'm mm-hmm. going to do my PhD there.
Right.
But you get that exposure, like you're saying, to different questions, different techniques, different ways of thinking.
Mm-hmm.
And I think this gets back to your point that if we have faculty that have different backgrounds, be that, whether we have a mix of men and women, we have different genders, we have people that come from different parts of the country, we have different races, like, you're going to have that diversity of viewpoints.
Yeah.
And that gives you these insights where you can apply these tools that maybe you never thought of yourself.
If your lab is a biochemistry-driven lab, and the guy next door to you is interested in changes in gene regulation, those are different aspects of a common problem.
Yeah.
And
they're clearly connected.
They're clearly connected. And so having multiple interactions on that level, I think, is really important.
Yeah.
Even if you don't want to do the genetic aspects of what you're doing.
Yeah.
Biochemical changes will change gene expression.
Yeah.
Gene expression changes biochemical actions. Yeah. So I think that having those kinds of interactions is really important.
Yeah, absolutely.
I don't envy you editing this because we've talked a long time.
Yeah, I'm going to have to see what to do on this, but I think I will let you get some lunch, because I think we've definitely worked up to another donut.
Well, there's a glazed donut with my name on it. So
let's call it a break for this one.
Okay.
So thanks again, Judy.
Oh, it was my pleasure. It's good to talk to you, Jim.